Okay, video's going. All right, Kendra, stop texting. I'll cut that out of the video, maybe. So I'm going to teach you about inotropy, lusitropy, and chronotropy within the heart through sympathetic and parasympathetic innervation, specifically by use of the beta 1 and M2 receptors on your heart. Really important. And then I'll also talk about like concentrations of calcium, like the question says. Whoop-de-doo. We're going to start with the beta 1. So Kendra, pay very close attention. So the beta 1 receptor, the one thing that we are supposed to remember about it is that it will always increase CAMP. The way it does that, so the alpha subunit pops off because it's a G protein, and that will activate a protein called adenylyl cyclase, which will start taking AMP and turning it into cyclic AMP. That cyclic AMP will then activate something called protein kinase A. That's that PKA. PKA sticks phosphate groups on whole bunches of things. And so for the stuff that we care about that will mess with your heart, it's going to phosphorylate these 4 things. We'll start here. The RYR, so your ryanodine receptor channel on your sarcoplasmic reticulum, little channels that let calcium out. So it phosphorylates them, that means they're more likely to be open. So you have an increase of calcium into the sarcoplasm, which means you have more calcium to bind, which means you have more myosin heads binding. So stronger contraction strength, which is what your inotropy is. Great. Got it? Mm-hmm. Okay. Next thing that it does is it will phosphorylate troponin I. So you've got, where are you going? Oh, you're moving. Okay. Ooh, getting closer. This is great. So you have the actin filament, then you've got the tropomyosin that sits on top of it, and then you've got the little Mickey Mouse troponin that's on there. So when you phosphorylate the troponin I, that means the calcium on the troponin C has a lower affinity, so it's more likely to just fall off faster. So if that falls off faster, that means the myosin heads will get knocked out of the way because the tropomyosin is going to come back in. So those come in, myosin head pops off, and that happens faster, which is why you can relax the heart faster. Make sense? Great. Okay. Then we're going to talk about phospholambin. Phospholambin is really weird. Still not quite entirely sure exactly what it is, but what it does is it makes your circopumps act faster. So you're taking calcium that's in the sarcoplasm and putting it into the SR faster. And because you're doing that, that means your calcium concentrations inside the cell are decreasing. Because they're decreasing, you have less calcium to bind to the troponin, which means myosin heads are leaving faster, which means your speed of relaxation is increased. What this also does is because you're putting more calcium in the SR, when you open the ryanodine channel, you can increase the amount of calcium you pump out, which will also increase how strongly the heart contracts. So 2 things there. Then now the last thing that it does is it interacts with our calcium, the L-type calcium channels. These are voltage gated channels. Phosphorylates them, that means they're more likely to be open. Calcium rushes in, depolarizes heart, contracts, whoop-de-doo, more calcium, more myosin head binds, stronger contraction. Increased contractility, otherwise known as inotropy. These ones, those are called lusitropy. Cool.
Somehow. Perfect.
Great. Any questions?
Nope.
Great. All right, so what were to happen if I were to decrease calcium concentration outside of the cell?
Outside of the cell?
Mm-hmm.
It's not going to cause it to contract, right? Like if we're talking for the RYR, because calcium goes in.
From where? That's a good thing. We'll start, if we just look at this one. Calcium is coming from where? This is coming from the SR.
Yes.
That's inside the cell.
Yes.
But if I decrease concentrations outside of the cell.
It's not going to do anything with that.
Exactly.
Okay.
Does that make sense?
Trick question.
No, it's not. That's just what's happening here.
The SR, yeah.
Okay. Does that affect anything here?
Mm-mm.
No. Anything here?
Mm-mm.
No. What about here?
L-type calcium channels are on your membrane?
Yes.
Okay.
So
Wait, what was the question?
if I decrease calcium concentrations outside of the cell.
Outside of the
So
cell?
here's a cell. Here's our.
You're going to have less contractility. Less contractility.
Less increased contractility.
Less increased.
So it's going to, instead of increasing by like this much, it's going to increase by like that much. It'll just
Okay.
increase slightly less
Alright.
because I have less of a gradient of calcium rushing in.
Okay.
Does that make sense?
Mhm.
Great. Good question. Where's my eraser? Here it is. Okay, now we're going to talk about what makes your heart beat slower. These are called chronotropy. Anything that increases your heart rate or decreases your heart rate is chronotropy. Chrono, time. Cool. And two, those are the ones that are usually on your heart. That's your parasympathetic innervation, acetylcholine, rest and digest. You don't need your heart pumping a whole lot because you're like, so that's activated. The alpha subunit is going to go and end up decreasing CAMP. Essentially, from what I understand, that decreases how much adenylyl cyclase is active. So you have less CAMP, pKa is less active. If pKa is less active, that means your L-type channels are not being phosphorylated, which means they're not open as often, which means you have less calcium coming in. If you have less calcium coming in, you have less contractions, less depolarizations, your heart rate goes down. Another thing, if CAMP decreases, in your autorhythmic cells, so your SA node, AV node, bundle fibers, and your Purkinje fibers, they have the funny channel, which is either hyperpolarized, so under threshold, or if a cyclic nucleotide is bound to it. If I have less cyclic nucleotides, that means I have less cyclic nucleotide, let's say CAMP. If I have less CAMP bound to the receptor, it's not open, which means I don't have as much stuff coming in, doesn't depolarize as fast. Hence, decreased slope. If I draw, we'll draw this. That's threshold. So normal autorhythmic cells will be like this. But if I decrease CAMP, this isn't opened as often. Decrease slope, so it'd be more like this. Like kind of like that. So this is happening less often, heart rate goes down. And then the fun thing about this one is so the beta gamma subunit of the G protein will also dissociate and it's going to open the GERK channel. And so the fun thing about GERK stands for gated G protein inward rectifying potassium channel, which is a really, really fun name for saying if it opens, potassium leaves. Potassium leaves, it hyperpolarizes. So that lowers the resting membrane threshold. So it goes down here. And so the funny channel just takes longer to hit threshold, hence lowering your heart rate. Cool?
Very good.
Great. So if I were to, say, decrease the intracellular concentration of potassium or increase potassium concentration outside. So here's my cell, and let's say I've got like 100 to 5 instead of the normal 150. So it's no longer 150, now it's 5 on the outside for potassium. If I open the GERK channel, potassium leaves. That's what that means.
Mm-hmm.
If I drop concentration out here, what does that do?
The driving force is lower,
Driving force is lower,
so you're gonna have less positive leaving.
which means what to my resting membrane?
It's gonna go up.
Which means what happens to my heart rate?
It increases.
So it decreases less.
Okay.
Important distinction. So if I had the normal amount, say my resting membrane would be here. If I open GERK, maybe it dropped down here. If I have less potassium leaving, it's still going to drop, but it's not going to drop as much.
Okay.
Does that make sense?
Yeah.
Okay. And the other thing that I'm supposed to talk about is calcium, but calcium is really boring. That's a lie. Calcium's really important. But the question asks, like, how does calcium move in and out of the cell? In your autorhythmic cells, you have the sodium-calcium pump. Calcium moves out, sodium goes in, woohoo, anti-active transport, secondary active transport. And then also uptake by circopumps, move it into the sarcoplasm, So you can remove a lot of the calcium from inside the sarcoplasm. And that's it. Thank you.